Hi, all RNA nucleotides. In the lecture today, we will start from long time ago, when the first cell appeared, when DNA, RNA nucleotides, ribosome, had not yet figured out a proper way to cooperate. That day, Mr. DNA received a letter from Miss Ribosome. Mr. DNA blushed at the time, but after a closer look at the letter, he found he was too confident in his charm. Miss Ribosome was asking for help in making protein. DNA was so embarrassed and would feel ashamed to see Ribosome again. At this time, he saw some nucleotides playing a set. Adenine connected to uracil, cytosine connected to guanine. He suddenly thought of a good idea. Mr. DNA wrote the required sequence for building a specific protein on the blackboard and asked RNA nucleotides to stand in the corresponding order following the complementary base pairs. The RNA nucleotides in the queue were sent to Miss Ribosome, and she quickly understood the meaning of DNA. With the recipe made by RNA nucleotides, ribosome began to build proteins. This is the very first recorded transcription. We, as nucleotides, must complete this course if you want to become a member of RNA. This video will introduce the transcription mechanism and difference between transcription in bacterial cells and eukaryotic cells, since I know you are aiming to different faculties. Before the lecture starts, I recommend you to pause the video and do these review questions. Transcription is basically the first step of gene expression. The DNA sequence of a gene is copied into RNA. Transcription happens on DNA, which is an antiparallel double helix, consisting a coding strand and a template strand. RNA nucleotides are the building blocks of transcription. The main difference between them is the bases. The main catalyzer is RNA polymerase, an enzyme that synthesizes RNA from a DNA template. The job of her is to match the complementary bases. She would allow A pair with T, C pair with G. Notably, RNA polymerase only reads the template strand in the direction of 3' to 5' and synthesizes new RNA 5' to 3'. Before transcription can take place, the cell needs to solve the problem that DNA is a double helix. Helicase is the enzyme that unwinds the strands. She's like a diver who cut off the base pair C weeds in the way of DNA polymerase, forming a transcription bubble. Then, initiation of transcription would start. There is a promoter sequence on both strands of DNA, while only one of them is served as the template for RNA. The direction of transcription is from promoter to terminator. On the other hand, RNA polymerase only reads the DNA in the direction of 3' to 5'. So we combine the two criteria. We find that in this case, the bottom strand is the template strand, where RNA polymerase would bind on, starting moving right, synthesize RNA once it meets the start site. By the way, the start site is usually located several bases downstream from the promoter. It is drawn at the first base only for simplification. As RNA polymerase moves along, complementary RNA nucleotides are added to the strands. The extension of RNA strand is called elongation. When RNA polymerase meets a termination sequence downstream, boom, she dissociates from the strand, and transcription is terminated. This is the common process happening in both bacteria and eukaryotes. Now, let's look at the difference between transcription in bacteria and eukaryotes. Starting with initiation, there are two promoter sequences in bacteria, namely negative 10 box and negative 35 box. They are named after their relative position to the +1 site or the start site. Negative 10 box is approximately 10 bases upstream from the start site. They are negative because we consider the direction of transcription as positive. Besides, RNA polymerase herself can only make weak connections to the DNA strand, which leads to low transcription level. Therefore, there is a helper protein called sigma factor who helps her bind to the promoter. RNA polymerase together with the sigma factor is called holoenzyme. Sigma factor wouldn't stay long. He dissociates from DNA as soon as transcription is initiated. In eukaryotes, the significant sequence in promoter is Tata box, around 30 bases upstream from initiation site. Similarly, an enzyme called Tata binding protein recruits and helps RNA polymerase bind to promoter. Meanwhile, there are quite a few initiation factors binding along. This group of molecules are called basal transcription complex, which also dissociates when transcription is initiated. And we proceed to elongation. Elongation is the same for both bacteria and eukaryotes. RNA polymerase would extend the RNA strands from the start site, making complementary base pairs with DNA bases on template strand. You may notice that the bases on the newly synthesized RNA strand are all the same as the bases in coding strand, except that the T bases in DNA are changed into U. Last but not least, the termination of transcription. In bacteria, there are two types of termination, role-dependent and role-independent. A role factor is an enzyme which terminates transcription. In a role-dependent bacterial cells, there is a sequence in their DNA which encodes the role binding site for RNA. That's where role factor binds to. Role factor would then move along the RNA towards the 3' end and unwind it from the DNA. And so transcription is terminated. In short, role factor does the same job as helicase. In role-independent termination, transcription is terminated due to specific sequences in the DNA template. You may notice that C and G nucleotides have 3 fingers, so they can make 3 H bonds with each other, while A, T, and U can make 2 H bonds. That means CG bonds are more stable than AU or TA bonds. As RNA polymerase approaches terminator, it hits a region rich in C and G nucleotides. The RNA transcribed from this region folds back on itself, and the complementary C and G nucleotides bind together. The result is a stable hairpin. Following the hairpin, there is a region rich in U nucleotides in RNA. AU bonds are relatively easy to break, so RNA strand is released. In terms of termination in eukaryotic cells, it is a bit more complex. In fact, termination processes in eukaryotes are not fully understood now, since termination processes are different from one another with different RNA polymerases. These are only for your information. In RNA polymerase 1, termination factor binds downstream of the RNA coding region, causing the release of the RNA polymerase and the new RNA strand. In RNA polymerase 2, the 3' tail on the ending of the strand is bonding at the polyadenylation site, but the strand will continue to code. The newly synthesized ribonucleotides are removed one at a time. RNA polymerase 3 terminates after a series of uracil nucleotides in RNA because of the weaker bonds between U and A. Unlike in bacteria, a hairpin loop is not required in eukaryotes, but it may enhance termination efficiency in humans. Eventually, RNA strand is released from DNA template. Is it ready to go out of the nucleus? Actually, no. The RNA strand we just made are only primary transcript. In order for it to become mature, it has to go through RNA processing, which contains 3 parts. First, a cap is added to 5' end of RNA strand. The end of the primary transcript are particularly susceptible to a class of degradative enzyme called exonuclease. The added 5' cap uses an unusual linkage between nucleotides, which are not recognized by exonuclease. The 5' cap also aids in transport out of the nucleus and helps bind the mRNA to the ribosome. Second, polyadenylation happens at 3' end. To protect the 3' end against exonucleases, a poly-A tail is added by poly-A polymerase. Poly-A is a chain of adenine nucleotides, 100 to 200 units long. The poly-A tail is susceptible to nucleases, but it does not have any protein coding functions, so it does not particularly matter if some of the A residues are degraded. In other words, poly-A tails sacrifice to keep the protein coding portion of the mRNA remains intact. The most striking events in RNA processing occurs because the protein coding region in eukaryotic genes is not continuous. It is composed of protein coding regions called exons, separated by non-coding regions called introns. The process by which introns are removed and exons are joined to one another is called RNA splicing. Some primary transcripts can be spliced in a few different ways. Such alternative splicing yields a range of related proteins. After the processing is complete, eukaryotic transcription officially ends. The mature RNA will be transported through nuclear pores to the cytoplasm where translation will occur. After the lecture about translation next week, you will be graduate. All RNA nucleotides watching today, hope you will do well in your positions. See you next week. Oh, remember to do these exercise questions. Thank you.